Good morning. Welcome. My name's Steve Schaffner, and is the sound good? Okay. I'm standing here because I'm talking today. I'm a computational biologist. I've been kicking around the Broad since before it was the Broad, so I've been around for a while. I was, for a number of years, I was a regular in MPG. I used to work for David Altshuler. These days I work more on pathogens, so instead of going to MPG meeting, I go to the malaria meeting on Thursday mornings, but I'm still interested in these issues. And what I'm talking about today is natural selection and what effect it has on human genetic variation. What natural selection is pretty well known. It's a very simple principle that the alleles or combinations of alleles that make an organism more likely to survive and reproduce are also more likely to be passed on to later generations. This turns out not to be always true when there are qualifications and caveats and complications, but as a principle, it's a simple principle, and understanding that is you understand most of adaptive evolution and sort of the core of Darwinian evolution. It's convenient to break it down into different types of selection, and the first of these is balancing selection, which is the least common. It's a selection that maintains multiple alleles in the population. Purifying selection is the most common, and this is selection that it selects against a new trait that's deleterious. And positive selection is the converse of that. If you're selecting against one thing, you're selecting for something else. It's selection for a trait that, and you usually think of it as something that's new or lower frequency, and that's being selected for to spread in the population because it gives an advantage. And I'll talk about each of these in order. Balancing selection, as I said, is not very common, but it happens to be the classic example of selection in humans is balancing selection. That's the sickle cell trait. That I think was the first clear instance of natural selection that we could see in humans. It was 1948 that Haldane observed that pathology of hemoglobin in various places around the world, the locations where it occurred tended to be places where there was a lot of malaria. The top map shows where malaria is endemic. This is falciparum malaria, historically where it was endemic. And the bottom map shows where the sickle cell allele occurs at high frequency or any frequency. And there's a strong correlation. And further investigation showed conclusively that the sickle cell allele does in fact give protection against falciparum malaria, although the exact mechanism is complicated. But you can in fact observe this directly. This is from some historic data set, I'm not sure which one, but comparing the survival of people, of children, with who have either the wild type allele or one or two copies of the sickle allele. And the top line is heterozygotes in blue. These have the best survival probability because it does give protection against malaria. Malaria is a big killer when there's not a lot of treatment going on. Second best is to have two copies of the wild type allele. You get malaria, may kill you, but otherwise you're healthy. The worst is to have two copies of the sickle cell allele, in which case you probably have sickle cell anemia, which is a severe disease, high mortality if it's not treated with in fairly sophisticated ways. So what this is it's called heterozygote advantage. The best, in this environment, the best thing to be is to have one copy. Your chance of survival is highest. Obviously, this varies a lot with environment, whether this is actually selected for. If you move out of that environment where there's a lot of malaria, or the environment changes and you start treating malaria, suddenly this becomes a deleterious mutation, or deleterious variant, and is selected against. It also, incidentally, shows that natural selection is not always nice. It's kind of brutal. So up to, in some areas, 2.5% of births are homozygous in some places for the allele. So there's a real cost. If you're one of the people who's being selected against, it's not necessarily fun. And that's one kind of balancing selection. There can be other kinds of balancing selection. Selection, not because the heterozygote is necessarily advantageous always, but selection for being different, especially being different from those around you. This can be, in the case of predators, if predators are keyed in, hardwired to look for a certain trait, and you can change that trait, you happen to have a slightly different version, you may escape from predators. It's a big deal in infectious disease, because if a new pathogen moves into your town and all your neighbors are getting sick, it helps if you're different and if you look different. So it's some different viral type might be easier to infect you rather than your neighbors, so you want to be different from them. So in general, this acts to increase diversity in the entire population. And the classic example of this is the HLA region on chromosome 6 in humans. It's critical for the immune system, and different HLA types are better at fighting different pathogens. And so if you just look at the number of SNPs at different places along chromosome 6, this is just a plot of that, right at the HLA region, there's a huge excess because there is strong selection pressure to have lots of diversity in this region. As a side note, oh, by the way, this is an example of frequency-dependent selection. The environment may not change, but whether something's advantageous depends on what your neighbors have. So it becomes advantageous if it's rarer. As a side note, I'll say, while we're evolving, our immune system is under this selection pressure. Pathogens, of course, evolve as well. And so any protein that's directly exposed to the immune system is likely to be highly diverse. And this is one such protein. This is in falciparum malaria. On chromosome 8, there's a gene here that's exposed to the immune system. There's a tremendous amount of diversity there. There are also actually multiple copies of this gene. They're all highly diverse. And so while we have evolved to have diversity to handle pathogens, they evolve as well, and of course they evolve faster than we do. So it's a little bit awkward. Okay, so that's all I'm going to say about balancing selection. It's hard to detect. It doesn't happen that often. There are various searches for it that have been done. But beyond the few classic cases, it's a little bit controversial as to how much has actually been found. Purifying selection is the bulk of natural selection that happens, especially in humans. It's by far the most common because it's a lot easier to break something that works than it is to improve it, as if you're just doing random changes. So if there's a change to a protein or even a regulation of a protein, it's more likely to be deleterious than not. And it is common. On average, every person is born with one or more deleterious mutations. The average number is somewhat higher than one. Exactly how much higher is not quite clear, but it's happening all of the time, and these mutations are being selected out all the time. And this also means it overlaps with medical genetics because some of these things are they're deleterious by causing disease or various medical conditions. It's easy to see the effect if you just look at genes across the genome. This is from 1,000 genomes data, and they've summed across all the genes. And here it's broken down into upstream of the gene, the first coding exon, first intron, and what's measured here is just genetic diversity across 1,000 genomes population. And you can see that where in the coding exons, there's a marked decrease in diversity. And what this is the mutation rate is the same there. It's just that a lot of mutations that have happened there have been selected against over the course of time. So over the past 500,000 years, the mutation has occurred and then been eliminated by natural selection. There's no guarantee that these mutations will be eliminated immediately. Obviously, some are not viable at all, in which case you don't see them, but they can stick around for a while. You can have a mutation that makes you more susceptible to disease or less attractive to a mate or something, and it can stick around. You can pass it on, but you're less likely to pass it on. And so if you look at allele frequencies, mutations that are more likely to be functional, more likely, and therefore more likely to be deleterious, tend to be at lower frequency. So what's plotted here is the allele frequency spectrum for non-synonymous SNPs in red and synonymous SNPs in blue. And it's very similar at most frequencies, but at the lowest frequency bin, there's an excess of non-synonymous mutations. And that's because these are mildly deleterious alleles that will be eliminated eventually by selection, assuming the selective pressures don't change. You can break it down in other ways. You can look at it in more detail. Here it's broken down into non-synonymous changes that are likely benign, that are because of the amino acid change, they're unlikely to change the protein structure. Those are shown in green. That's the second column there. And there's not much of an excess there. But ones that are probably damaging, probably change protein structure, there are ones that are likely to be deleterious. And so these two columns here are the ones that you have an excess of those. So these are in the population. They're contributing to disease burden, and there's some of the things that you look for in GWAS. So that they tend to be at lower frequency. Purifying selection affects not only the variant itself, the mutation that happens that might cause a problem, but also affects the diversity in the surrounding area, because when a mutation happens, a deleterious mutation happens, that chromosome isn't going to last for a very long time. It happens in a particular chromosome, it might get passed on for a few generations, but it's effectively lost from the population. So around genes, some number of chromosomes just keep getting lost from the population. And that's similar to having a smaller population size just right around that gene. And so the effect is there's lower diversity. There's just less opportunity to accumulate diversity right around genes. This is called background selection. And again, it's easy to see. You just look at the total diversity in a broad region around a gene. This is 1,000 genomes data again. You can see that there's a marked decrease in diversity right around a gene. Okay, and that's, for human health, purifying selection is probably the biggest thing, but in terms of like evolutionary biology, we don't care much about it because these are just things that pop up and then go away again, and they don't lead to cool, sexy, new adaptive traits. So we tend to think more about positive selection if you're sort of in an evolutionary kind of frame of mind. Yeah.
One question. Given that we don't know many mutations in the non-coding regions, but increasingly we're finding them, does this track, in fact, with mutations that you see? Does it would look like they're regulatory mutations that are adaptive rather than coding mutations from what you're just saying? But has anybody now gone back and mapped these things on this area to see if that's really true in terms of distance from coding sequence, et cetera, to get some insight into what are the regulatory ones that are going and where they might be introns, exons, 5' end, the way you did on the previous two slides?
Are they looking for beneficial mutations or
either?
There have certainly been searches, and you can see the effect of purifying selection on regulatory regions. It's not, it's weaker because the regulatory regions tend to be sloppier, there's often redundancy, but there is a signal there, and there's also signal, or at least it's been claimed that there's a signal for positive selection happening at regulatory regions, and most positive selection does happen at regulatory regions as far as we can tell. So yes.
Is there any preferential site for those 5' end introns? Do you gain any information evolutionarily from where they might be adapted?
You can, oops, go backwards here. You can, even from this, you can see that 5', if you compare, I actually don't remember, 5' or 3, I think it's 5', it tends to be more, show more sign of purifying selection. I think there's more going on at 5', but I'd have to check, but I believe that's the case. Yeah, you can indeed see that.
As we have more whole genome sequences, we'll be able to map this better.
Yes, more whole genome, yes. And
just wondering, where's the action in the non-coding sequence?
Oh, yeah, yes. There's a great deal of work going on now in understanding exactly where the action is. And because we don't always know, even when we know selection has happened somewhere in this region, we don't necessarily know exactly where. It's sort of a conversation, or collaboration between looking for the selection and then all the stuff we're doing now to figure out what's actually regulatory and what's important in regulation. I'll mention a little bit of that later. Okay, so I'm going to talk about positive selection now. In particular, I'm going to start with positive selection that's starting from a new mutation or a very rare mutation, because it's like sort of the classic ideal case. You start with, you have a population, and there's a new mutation that makes you better, and that then spreads in the population. So the idea is if there's no selection going on, all right, you have a blue baby born, and the blue baby has no advantage, and the frequency of blueness may, go up and down a little, but it's basically just, it's not going anywhere in particular. It's neutral evolution. On the other hand, if a red baby is born and being red is good, well, then it increases rapidly in frequency and spreads in the population over a fairly small number of generations. And it's this rapid rise in frequency that produces signals that something has been going on here. It changes the genetic variation in the population and means that we can then test for whether one of these things, this is called a selective sweep, whether a selective sweep has occurred in the population. So the idea is that initially you have a bunch of different haplotypes in the population, different genetic variation, and this redness mutation occurs, and then afterwards it's spread in the population. And now in the region around that particular change, everybody's the same, or at least most people are the same, depending on how frequent it has become. And that's basically what you're testing for. One thing you can test for is to see, suppose if this has happened in one population, like in West Africa, say, then people there have a lot of copies. A lot of people have the red copy now, and people elsewhere don't. And so you just look for alleles that are at high frequency, much higher, unusually high frequency in one population compared to other populations. And it's a simple test. It's often measured by FST is the statistic. There are other tests. And there are some the classic case of this one is the Duffy null allele. The Duffy, it's a protein, surface protein on red blood cells. It is the way by which vivax malaria parasites enter red blood cells. So if there's no Duffy on the surface, there's no entry, there's no infection. There's a mutation that occurred in Africa that removes, it removes expression of Duffy and provides almost complete protection against vivax. And as a result, and you can see, you can see from the map here where the Duffy null, it's called the Duffy null allele, where it occurs. It occurs in Africa where there once was a lot of vivax malaria. It was so effective that vivax malaria was almost completely wiped out from sub-Saharan Africa, although it may be coming back in again. Apparently there are mutations in the parasite. And there's also some indication there may still be a little bit of vivax malaria, and it's not entirely clear why. So there's some other little subtleties to the story we don't quite know yet, but basically it's a simple case of highly successful evolution in humans. And this kind of signal has been used elsewhere. This was a nice study from, when was this, 2010, looking at allele frequencies in Tibet. In this case, comparing allele frequency in Tibet on the x-axis with the frequency of the same allele in Han Chinese, who are a very closely related population. And there were some outliers. They were looking for outliers for differences. And down here you can see derived alleles that are high frequency in Tibet and much higher frequency in Tibet than in the Han Chinese. And these mutations, it turned out, with further investigation, confer adaptation to high altitude, to being able to handle low oxygen levels. And they're just, they stick out like sore thumbs, very clear. And so it's a way of finding what's been selected for in a particular population that has a different environment. Yes?
I mean, if you have like structure, like events like bottlenecks, then a sudden increase in allele frequency can also happen just by chance, right?
Yes, and if you have a bottleneck population, the frequencies change, but all of the frequencies, frequencies in general will change, and you'll just see this distribution be much wider. And so it would be very hard to pick out. It may not work in the case of a bottleneck population. But it's basically how wide is the distribution in general? What's the neutral distribution? And then are there outliers? And in reality, there aren't many cases where there's a clear outlier. You can see in this comparison, there are the ones down in the right-hand corner, and there are a few in the upper left-hand corner that might be interesting. But mostly it's, they're all pretty consistent. And there are very few cases, the Duffy null allele where it's at 100% in one population and 0 in most of the world, that's very rare. There are only a handful of cases known. So it's a very powerful signal when it happens, but humans have not, we haven't been separated that long from each other that this has happened very often. I think I'll skip that. Basically saying what I just said. Now, all right, so that's one signature that selective sweep has happened. Another signature is very simple, that there's low diversity. If most people in the population have the same same haplotype in a region, they're not very different. You just measure diversity across the genome. You're going to find places where there's low diversity. The only trick here is you have to take into account that mutation rates also vary. So you may want to have some estimate of the mutation rate, like by comparing to a different species. So that's a signature. Then you can also just look for the haplotype itself. Look for long haplotypes. Look at those long correlations. So if you have phased haplotypes, in these days we have large data sets of either sequencing or genotyping where we've done phasing, and so we have a good idea of which alleles go together. And if you keep seeing the same haplotype over and over in lots of people, and it is not broken up as you move out from an allele, that's a sign that this has been well, it's a sign that this is a very young region, that recombination hasn't had time to break it down. If it's at high frequency in the population, that means positive selection has probably been happening because it's young and it's frequent, and that means something had to make it frequent. This is a very powerful test in some cases. This was from HapMap data, so this is fairly old. And if you compare, plot on the x-axis in this plot is the frequency of haplotypes, and on the y-axis is the length of haplotypes. So the locus that had the longest haplotypes at high frequency is this red one up here, which occurs around near the LCT lactase gene. And it turns out there's a mutation there that confers lactase persistence. That is, as an adult, if you come from Northern Europe, you can probably digest lactose and you're lactose tolerant. That's not the sort of normal mammalian condition. Similar mutations have occurred in other cultures, other populations that have a tradition of herding because they had milk available to them. And this was, by this test at least, this was the strongest signal for selection in Northern Europe. It was very powerful, selective advantage of something like 10%. So it clearly provided a lot of advantage being able to drink milk, at least at some point in the past. Okay, so the final signature that can happen, thing to look for, just if you're looking across the genome, where a selective sweep has happened, is you may find an excess of high-frequency derived alleles. That is, the new mutations arise as a single copy, so the derived allele, the non-ancestral allele, tends to be at low frequency. But if a sweep has happened, it can sweep up a bunch of stuff that was at low frequency previously, and you tend to find an excess of high-frequency derived alleles. So that's the last of these classic tests for selective sweeps. And people took these tests, and they went and when we had whole genome data, finally, we took them and went and looked for where selection happened in the genome. You can then just go across the whole genome and look, and you can scan. And you can also do a little bit more intelligent things, like these are different signals. Well, why don't you just combine them and look at more than one of them at once? You can look at each one individually, but an approach here adopted by Party Savetti at the Broad was, she called it composite of multiple signals. You just combine them in a single statistical test. And you combine the information about long haplotypes and derived alleles and differentiation mean populations, and you just statistically combine them and see try to narrow in on what variant was actually responsible, or what particular gene was actually responsible. Because the thing about a long haplotype in particular, it's great. It means that selection has happened here, but it's long. And it can cover a megabase or 500 kb, and it can have multiple genes and lots of variants in it. So zeroing in on it is quite important if you want to have any idea what's actually going on there. And here's one example from data. Each of these rows is a different statistical test for selection. They're all showing elevation in this region. So there's positive selection probably happened here. There was a selective sweep. You can see they're very noisy. And 3 of them are different long haplotype tests. The second one there is FST, the population differentiation. But if you combine information from all of them, you get the bottom plot. You can see it, where basically there's no signal across most of this region, and then right over here, there's one variant that's really looks a good candidate, and a few other not quite as good candidates. That particular one, that high probability one happens to be a non-synonymous change in a gene. The gene, this MAP-P, is known to be involved in pigmentation. This is in fact a mutation, or very likely was a mutation that conferred lighter pigmentation on people that had left, humans that had left Africa and moved farther north, and for whom it was beneficial to have lighter skin color. So that was great, and using this and various tests, lots of people did, a fair number of people did, scan the genome for instances of positive selection, and produced all sorts of, maps of places where things had probably happened. Some of them covered genes, various regulatory regions, and it was cool. But then the question was, well, now what do we do? And the first question is, what do these things do? We have no idea. Unlike a GWAS, where you're looking for an association with a particular trait, we don't know what the trait is here, other than it was beneficial. And so, some of this is still going on. I don't know if you noticed, a lot of the papers I was, taking figures from were from like 2008, a little while ago, 10 years ago, say, when scans were a bigger thing. And because you get to this point and then, now it gets difficult because we have to figure out all right, so there's a mutation somewhere in a region, and now we have to start doing biology, and that's slower and harder, and less fun for some of us. So I think it became less of a focus, but it is still possible. It was possible to do the work. It was just a lot of work. Like, here's one example of doing the work, of figuring out what's going on, of taking a candidate. This is a candidate region on chromosome 2. This is a signal for selection in the East Asian population. And this was a long haplotype, like 800 kb. There was evidence for selection in this region. If you did what I said before, combined different signals, it made things a lot better. There were only a handful of candidates then, those few black dots. And one of those, again, was a non-synonymous change. In this case, probably likely to be a change that actually changed the protein and therefore changed function. Most of these would not be in actually in coding regions. Most of them are regulatory changes, but this one's, because it's a coding change, is the easiest one to look at. So somebody looked at it. Actually, somebody affiliated with Yana Kambarov, affiliated with the Broad, but now at UPenn. And then this mutation is in a gene called EDAR. It's a gene that was known to play an important role in skin development, in particular in development of hair follicles and sweat glands. It's also been under this pathway has been under repeated selection in other species. And so at that point, it required doing real biology and actual people who know what they're doing in a lab rather than someone like me. And so, as I said, this was in Eastern Asia, and it was only at high frequency in East Asia and in the New World. So selection presumably happened there, and creating a mouse that had this mutation showed several different phenotypes. It produced thicker hair, this derived allele, apparently a beneficial mutation, smaller and denser mammary glands, and more denser sweat glands, eccrine sweat glands, which are the primary cooling sweat glands. Just guessing, the sweat gland one is the most likely one to have been under selective pressure, because it's an important role in thermoregulation. We don't actually know which of these was selected for, or if there conceivably could be some other effect we're not aware of. But at least it's given that how much effect this single mutation has on a number of traits, it's reasonable to guess that this was in fact the mutation that was under selection. May have had something to do with thermoregulation at that time, and that's about as far as we can go in sort of reconstructing the history. But that was a substantial amount of work there. So that's kind of where we were with selective sweeps. One question that came up was, all right, if we have all these maps of where people claim selection has happened, the maps don't overlap all that well if you look at different people's claim of where their selection has happened. And the question is, how many selective sweeps have there actually been in recent human history? Or is it worth looking for these? You can say, well, selective sweeps reduce diversity. Well, so does background selection, as I said before. Just purifying selection reduces diversity around genes. So somebody came up with a clever idea. This was Ryan Hernandez and I think Molly Przeworski. And that's just compare diversity around non-synonymous mutations versus synonymous mutations. And these are mutations that are fixed, that have occurred between the common ancestor of humans and chimpanzees. At some point in history, there was a mutation, either a synonymous or a non-synonymous mutation. And some of these will have happened recently enough that if there was a selective sweep around it, we should see a reduction in diversity. So on average, across all genes, we should, if selective sweeps have been common, we should see lower diversity around non-synonymous changes. And here they've plotted over all genes in the genome, they plotted the diversity in current human population around the different changes. So the site of the substitution is in the middle. In dark blue is the diversity around synonymous changes, and in red is diversity around non-synonymous changes. And they're basically identical. And there's no sign from this that there's been a lot of selective sweeps reducing diversity around non-synonymous changes. And their conclusion was no evidence for a lot of selective sweeps, so that this kind of classic selective sweep where one thing sweeps to high frequency, they've been rare. And they actually pretty much said in the paper, we should stop wasting our time looking for them, which was difficult if you were trying to get a grant looking to go look for them. Then, the review, the reviewers are saying, well, why are you looking for them? Hernandez et al. Just showed that there aren't many, which was troubling. And, the limit actually meant there still could easily have been lots that were worth looking for, but it still caused some trouble with funding. But then a few years later, somebody else came along, David Enard's group, and said, wait a minute, you didn't do that right. That was you did the wrong test. And why is a little bit subtle, but I like this story because it shows how easy it is, how confusing things can be. So think about two genes. One gene, gene A, is highly constrained. That is, any change to this gene is likely to be deleterious. And so if you look in comparison to chimpanzees, there are going to be very few changes between chimpanzee and human. And it also means there's going to be lots of background selection here because all the changes produce deleterious mutations, they get swept out, and so there's lots of reduction in diversity around this gene. Gene B is kind of a floppy gene. A lot of it doesn't matter for function. There can be lots of non-synonymous changes that are perfectly neutral, so you'll see lots of changes between human and chimpanzee. And you'll also see less reduction in diversity around it. And so when you do the test I was talking about, where you compare non-synonymous, diversity around non-synonymous versus diversity around synonymous, when you take a non-synonymous mutation, you're probably taking a mutation from gene B, because that's where the non-synonymous mutations have happened. And so you're selecting places where there hasn't been much background selection, and so that reduction in diversity isn't very big. And if you then compare that to synonymous mutations, which can happen either A or B, if there has been a lot of, if selective sweeps have reduced diversity around these non-synonymous mutations, it's going to be masked by the fact that this trough wasn't very deep to start with. So it's going to, if A and B look the same, it may be because you've just selected a biased set. And so their conclusion was that there's actually strong evidence for lots of selective sweeps. There's been a high rate of strongly adaptive amino acid changes. They also concluded, because they didn't just look at genes, they concluded that most sweeps have occurred around regulatory regions. Again, picking out which one, statistically they're averaging over the whole genome, but picking out particular ones is difficult. Which, as I like this, that's their conclusion. My conclusion from this story is that things are complicated, science is hard, and that just because a paper got published in Science doesn't mean it was right. There's a certain tendency to think, that's established and stuff changes. And of course, there's no guarantee that the second conclusion is right either. Maybe they did it wrong as well. But it's an ongoing story. It's an evolutionary story about evolution. Okay, so as I say, that's a lot of that's a little bit dated. It's still, these studies are still going on. They tend to be more focused, but people still look for selective sweeps. But I think a lot of the action now, and some of the more interesting stuff that's going on, is in looking at things that aren't, there's positive selection, but not selective sweeps. Because the tests I'm talking about assume that you have a mutation that arises as a single copy or very few copies, and sweeps to high frequency. And that means it's either a new mutation and it's a one allele that has a large effect. But lots of selection, lots of evolution is probably not like that. There's lots of standing variation in the population. As you move into a new environment or the environment changes, that can be beneficial, some of that variation. And that's not going to show up in these tests because it's already occurring on lots of different haplotypes. And so there isn't one haplotype that's sweeping to high frequency. Multiple different independent mutations can happen that can produce the same effect. Like the sickle cell mutation has actually happened multiple times and has been selected for in the same population. And a lot of selection may be happening for traits where there's no single allele that has a large effect, but this is polygenic traits where there can be more of a quantitative trait. There can be lots of small changes that can produce selection. So these are things that people are looking at now, and in various ways. And one important conceptual way of doing it is to look at a trait rather than an allele. Look for selection on a trait. And this is an early example of this. This was from 2012 from Joel Hirschhorn's lab looking at stature. And what makes this possible now is basically GWAS, because we now know lots of variants that are associated with lots of traits. And we're not interested just in the ones that were statistically significant, the handful of things that are really outliers. Now we can construct polygenic scores or risk scores based on a whole bunch of variants, many of which appear associated with the trait. Some of them may not be causally, but if you average over a lot of them, look at a lot of them, you're covering a lot of the genetic cause of that trait. And so what they did in this study was they looked at lots of variants that were associated with height in GWASs, and they correlated that with geography, where in Europe, these are mostly studies of Europeans, where in Europe their ancestors came from. And what they found was there were lots of small effects always in the same direction, was that Northern Europeans tended to have the higher frequency of the alleles that gave taller stature. Northern Europeans are on average taller than Southern Europeans. And because all of these things were shifting in the same direction, their conclusion, which was backed up by later studies, was that natural selection was operating either for taller stature in the north or shorter stature in the south or both. But that it wasn't that there was one allele sweeping to high frequency. It was just subtle shifts in lots and lots of alleles. Not surprisingly, stature, I mean, how big you are, lots of things could contribute to that, clearly. So that concept has been applied in various ways and is kind of core to a lot of what people are doing now. And another quite different kind of cool thing that we can do now is rather than looking at genetic variation now and inferring what happened in the past, we can actually look at ancient DNA now and see, did allele frequencies change over time? You can actually look at, rather than inferring, you can look at allele frequencies and you can watch them change over time if you have enough ancient genomes. And just in the last few years, we have gotten to the point where we can do that. This is from a study from David Reich's group, looking at 230 ancient, of the order of thousands to 10,000 years ago, roughly, genomes from Europe and Western Asia. And these are just examples of particular alleles that have been implicated as having been under selection, and just looking at different groups, different populations. The hunter-gatherers in green, some early farmers in blue, and some steppe populations, Asian steppe populations in red. Just looking at the frequency of these alleles. One thing you can notice just from the lactase mutation is basically absent during all of these early groups. It was actually a very late mutation that rose very rapidly, fairly late in European history. And what they did was they took these 230 genomes and just looked for evidence of particular SNPs that had risen to high frequency rapidly over this time. And so this is a different kind of selection scan than had been done previously. And the things that were found were similar to what was found with more traditional scans. Where we know what a gene does, There are ones clearly involved in skin pigmentation. There are ones involved in diet, like lactase, but also fatty acid metabolism. Ones clearly involved in infectious disease, and of course other ones where it's not known what they do, again. What was interesting about this was that it's telling a similar story to other ways of doing a selection scan, but the story, when we have more information, turns out to be more complex than what our simple population genetics models suggested. So I just told you about the stature, like there's this gradient north to south. But when you look at the ancient DNA, you discover there was evidence for selection for shorter stature in Southern Europe. There was evidence selection for taller stature in steppe populations in Western Asia, who then invaded Northern Europe. And it wasn't that there was necessarily selection for taller stature in Europe, it was selection elsewhere and people moved in. Because turns out people moved around a lot. And a somewhat similar story for pigmentation. One of the classic cases of selection of pigmentation gene that confers lighter pigmentation allele, an SLC45A2, has this sort of standard story. It was beneficial in Europe to be lighter. This allele rose in frequency in Europe. Another gene, which looks very similar in name but isn't, SLC24A5, had already been selected for in the Eastern Mediterranean among farmers, among the population in Anatolia. That's where farming came from. These farmers swept into Europe, displaced most of the existing population. And it wasn't that it was selected for in Europe, it had already been selected for elsewhere, and it was just this migration that produced the signature that we now see of selection having happened in Europe. Again, it doesn't fundamentally change what selection was for, but it means that it's an important message that when we reconstruct the past, we have these nice models, and the models are often wrong. That was particularly the case for EDAR, which I just described, that selection in East Asia. And you're just looking at the map. This is from the original EDAR paper. On the left is the allele frequencies in different populations, and the red is this selected allele, and it's present in East Asia, it's present in the New World. You know, it arose in East Asia, was selected for rapidly, spread rapidly, and as the peopling of the New World to the Bering Strait, it went into the New World. And they did some very complicated, sophisticated modeling, and on the right you can see where this allele probably arose. In northern China, some estimate of when it happened. But then when David Reich's group looked at ancient DNA and discovered this allele was also present at high frequency in Swedish hunter-gatherers 6,000 years ago, which is not what the model said. I mean, it was just wrong, which is fine. But, you know, and again, it doesn't really change the selection story, but it should change our confidence in our reconstruction of exactly what was going on in the past. So again, things are complicated, but that's good. All right, nearing the end. Switching from watching selection directly in the ancient past, look at in the present. It turns out we can also look at selection operating in the current generation directly, which is kind of cool that we can do this. Because now we have not only lots of genotypes and we have GWAS, we have sort of detailed biobank information on medical information and various metadata on humans in the current population, as well as genetic data. And so you can look at first thing you can do is look at changes in allele frequency as people age, what causes them to die, basically. And the things that are more protective, they go up in frequency with time. And you can do that either for individual SNPs or for, again, the polygenic score for a trait. You look for whether traits, genetically influenced traits, increase in frequency with age. And it's been done. This is from the UK Biobank. And in the upper left, if you can see, these are things that these are traits that are where the allele frequencies change a lot as people get older. And they're not surprising. TC is total cholesterol, LDL cholesterol, body mass index, coronary artery disease, big killers. It's not surprising that things that protect you become more common as you get older. The puberty timing is interesting. If you have later puberty, you're likely to live longer. Not clear why, but this might be an interesting biological story there. And if we just looked at the end point in time, this would basically be a GWAS for longevity, but instead they're actually looking at the progression over time at different periods. Now, most of these deaths are occurring later in life, and they don't have much influence on reproduction. So, the relevance of this for evolutionary selection, whether these variants will change in frequency over generations, is not clear. But I think it's cool that we can do it. And as we get larger and better data samples of younger people, we might be able to see more directly things that are influencing survival at earlier ages. But better is you can not only look at how long people live, but how many babies they have, what their fecundity is. And that's much more directly related to evolutionary success, because if you have more babies, then your genes are being passed on to more offspring, and they're more likely to spread in the population. And again, you can correlate how many, we know how many babies lots of people have had, and you can correlate what traits they have, what genetically influenced traits are likely to produce, are more common in people that have more children. And a paper that came out in 2018 has these results. This is showing the correlation between relative lifetime reproductive success, which is relatively how many babies you had, and various genetic traits. And what's interesting here is that, in terms of survival, a lot of the things we expect to see, to make you die earlier, like BMI, high cholesterol, high weight, also are associated with having more children. So this is split up by women and men, but you can see like BMI here in both women and men, I believe, yes, is strongly associated with having more children. Same with height. This is hip circumference, waist hip ratio. These are all things that we can think of as being not healthy, but evolutionarily appear in very recent, this generation or the immediate previous generation to be under positive selection, and exactly why is not clear. For some reason, height in women, shorter women have more children, not entirely clear. It might be a little bit disquieting to note FIS is fluid intelligence. So if you genetic factors that tend to confer greater intelligence are associated with having fewer children, which isn't exactly news, but it's a little disconcerting to see that intelligence appears to be selected against currently in the current population. It's, it is what it is. So a little summary here of what's been selected for. This was from a paper about a year and a half ago summarizing a lot of things, just different cases that have been, particular cases that have been seen. A lot of these I've already talked about, like lactase persistence, diet. And also, what is that one? Oh yes, lactase persistence has been selected for not only in Europe, but also in East Africa, probably in South Asia too. Some places. Skin pigmentation has been selected for in multiple populations. Different genes, some different genes in different populations, some shared. Stature, I talked about stature in Europe, which is a highly polygenic selection. Interesting, shorter stature was selected for in Central Africa and pygmy populations, but there it was not very polygenic. There were a small number of large effect alleles. So the similar selective pressures can operate in different ways. Is that equivalent? No? Stretching or questioning. High altitude adaptation has occurred on at least three times on 3 different continents, different mutations independently. And this is, I'm not quite sure I believe this, but this was from a paper recently, 2016, using a test that I haven't actually talked about. It's kind of a subtle test, looking for selection just within the last 2,000 years. And they looked at 43 GWAS traits to see whether there was evidence that there had been positive selection going on in those traits. And the larger circles here are ones that they have sort of confidence in. And like 13 out of the 43, or 15, something like that, showed evidence for selection. And some of them are, height, we've already said, fasting insulin might be a, BMI. We talked about some of these, but it's just sort of interesting that such a large fraction of the stuff of all of these traits seem to be under some selection. Not usually very strong selection, but their selection is ongoing. It's changing us constantly, and it's sort of related to health, and it's an important thing to understand. So I have no profound where are we? Profound comments, but basically, it's pretty clear that selection has been important in our recent biological history. It has effects in our health now, in our traits now, our biology now. It's one important tool for understanding what's going on, what has been going on, but we can only understand it in conjunction with lots of other different things. Ancient DNA, function, GWAS, these large data sets, understanding regulatory variation, because we can't understand what's going on in these sites unless we understand regulatory variation. So it's just one part of the entire package. And that's all I have to say. Any other questions? Feel free to applaud. Thanks.